So what I'm going to do here is look at some sequence files. These are sequences for H5 hemagglutinin that was downloaded from the NCBI in 2023 on the 17th of May. Doesn't really matter what the sequences are, they just happen to be an example file that I had kicking around. Now, first thing I'm going to do is have a look at this file using Notepad++, which opened in my other screen as is lucky. If you look here, you can see the names of each of them, and there's a lot of gaps in the titles in the headers. Now, if I open this file in GelView, which is automatically set to do, which you can see because it's got the GelView logo on it. So if I double click on it and open GelView, which again is opening in the wrong window, so here's GelView. Right. Now, if I make the sequence window up a bit larger so we can see things, and I slide this across to look at the name, the names of each of the sequences is just this first identifier. Now, that's not very helpful, because I'd sooner have all the rest of the information kept in the files when I'm reading it. Actually, if I slide down, I can see it one at a time, but it's still not very helpful, because if I want to know that's 2008, or I want to know it's an American black duck, or if I want to know it comes from Illinois, I'd prefer to see it here in the annotations, which it's not showing me in these headers, but it does if I glide down it. So I need to do something to prevent that from happening. Similarly, if I open this with Mega, so I'm going to open it to do an alignment in Mega, and again, species abbreviation, I get this. This is an inconvenience. So let's do a quick alignment. Line by muscle, go, okay. Hopefully I've not forgotten to check how many sequences I've got. Yeah, I'm not doing that. There's 2,000 sequences. No. Stop. Oh, yes, I don't want to do that. Now, the problem is when I do any derivative thing from it, I'm possibly going to end up with a file which just contains these codes. So if I want to look at the biological information which is there, which we saw before in GelView, is there, then I've lost it, and that meta information, that header, is really important. That's why I downloaded it. I wouldn't download it if I didn't care about it, because I might want to ask questions to do with which species does it come from, or what year, or what location. Any of these questions, if I've just got the IDs, then I need to go and find a reference table which then will tell me what each of those IDs mean. This is difficult and time consuming to have a look up. It's much better if I've got the file sorted out in the first place. So what do I need to do? Simple, really. So if I open this again with my editor, so I'm using Notepad++, so what I'm going to do is do a replace. And what I'm going to do in replace is I'm going to replace all of the spaces with an underline, and I'm going to press replace all. So now you see here you've got underline, underline, underline all the way across. Done. It's replaced it all the way through, and I can resave my underlined sequences. That's nice. And close the file. Now if I open it in GelView again, so here's my GelView, and now if I go to the full run, so now I've got the whole thing, so I can just look down this list of things and see the complete set of details, which makes it much easier for me to use and to do the analysis with. If I go to Mega and I do the aligns, and similarly in Mega, I've now got all that extra information, which means if I decide, oh well, I only want to look at green teal, I can select all select the ones that are green wing teal, I can delete the other ones. If I'm going to do any editing based on metadata, that's important. It's also important if I'm going to do any edits based on missing data, because it helps me to make decisions. If I find that there's 5 sequences that all came from the same location at the same time, and one of them has a lot of ends in it, so a lot of missing nucleotides which are incorrectly assigned, I don't really mind about deleting that. If there is a single entry from a particular location and it's a very rare species, then I'm going to be a bit more conservative about the things that I delete or don't delete. So having the full header information in the file is important. So the first thing to do with any of your files is remove the gaps so that you're getting the information through.